We now move on to the oceans. Oceans are another aquatic environment, but they have several important differences. First, they have ions at a concentration of 2.5%. NaCl makes up about 85% of the ions, but there is also sulfate, magnesium, calcium, potassium, bicarbonate, bromide, borate, strontium, fluoride, and others. Whew! Second, they are big. Oceans cover 70% of the surface of the earth, and because of this, surrounding land has much less influence on them in comparison to lakes. The ocean has abundant carbon dioxide. It also has oxygen in at least the top 20 meters that will be totally gone by 500 meters. Interestingly, because of downwelling, and that is just basically cold water sinking in the ocean, you will also find oxygen at the ocean floor. Nitrogen is also present. In all cases, you will find more gas in cold water than warm water. Light is also a key property that dictates metabolisms of the ocean. About 5% of light is reflected out of the ocean. The rest penetrates into the ocean, where it is absorbed by microbes, plants, and the water. Depending upon the number of particles in the water, the euphotic zone, that part that is exposed to light, can be 3 to 150 meters. So, who do you think would be the primary producers in the oceans? The answer is seaweeds and kelp, and mostly cyanobacteria. Beginning in the first decade of the 21st century, scientists began a cataloging of the Earth's oceans, using the tools of molecular microbial ecology to characterize the Earth's oceans. In the second expedition, called Sorcerer II, ocean water was collected on filters and the microbes were captured. All of the DNA was sequenced, so they were doing metagenomics, not amplicon sequencing, and assembled into large pieces of DNA called contigs. In other words, the sequence was all contiguous, right? It's not a chromosome, but there'll be large pieces, and then each organism will have anywhere from 20 or 1 contig to hundreds of contigs that make up its genome. That's not a chromosome, it's just the way we could sequence it together. Here's a more recent global analysis of the oceans. This data was extracted from the Tara Oceans Project, and you can see the link there. As you can see, the phylotypes vary depending upon the ocean location. The most common things that you find, surprisingly, or some of the most common, are chloroflexi, planktomyciota, alpha proteobacteria, cyanobacteria, and then the others. But you can see groups that you've seen before, but the proportions change depending where you are in the ocean. Going back to the Sorcerer II data, the results had many implications, but I'm going to focus on just one. 5 major genera make up the majority of the ocean: Pelagibacter, Prochlorococcus, Synechococcus, Burkholderia, and Shewanella. So now let's take a look at some of these organisms. A dominant organism in the oceans is Pelagibacter, a proteobacterium. One species has been isolated, Pelagibacter eublechi, and it has the properties listed on this slide. It is a oligotroph, and it grows on low nutrient densities. It doesn't like a lot of nutrients in its medium. It grows slowly. At 15 degrees, it took it 23 days to grow to stationary phase. It will only grow in seawater medium, so there's something in the medium that we don't know yet that it needs. It is a 1.3 mega base pair genome, which is relatively small. Remember, E. coli is about 4.5 million base pairs. It is a chemoheterotroph, and it has proteorhodopsin. So here we go again, that rhodopsin, and these rhodopsins are turning out to be pretty ubiquitous. It is also metabolically capable and can synthesize a lot of what it needs. Okay, so Pelagibacter is one example of a dominant organism in the ocean. And as you can see, it's relatively small. This bead shown here is about 1 micron wide, and you can see that these organisms are quite small. Cyanobacteria are also represented in the oceans, with Prochlorococcus and Synechococcus serving as important primary producers. Prochlorococcus are very small cyanobacteria, about 0.6 micrometers, and you can see a phase microscope picture, a phase microscope micrograph of them, right? And inhabit subtropical and tropical oceans. They specialize at growing at very low light concentrations and can grow to deep depths. Synechococcus are found in all oceans and are phylogenetically diverse, fresh ocean isolates and obligate photoautotrophs. They have cells that are about 0.6 to 1.7 micrometers and are ovoid. Again, you will find them in all sorts of oceans. So think back to our question about community physiology. What happens when you dump garbage into the ocean? What gets produced? Okay, if you think about it, if you're adding garbage to the ocean, you're adding carbon and molecules that can be depolymerized. Those will be depolymerized. They will go through this. Under aerobic conditions, you'll get water and CO2, but under anaerobic conditions where there is no oxygen, because there's sulfite ions in the ocean, you will get hydrogen sulfide, water, and carbon dioxide. This is why it's a really bad idea to dump our garbage into the ocean, and we should try to keep it clean, because hydrogen sulfide is a poisonous gas. It will kill ocean organisms, and it can also be dangerous to humans. Okay, the last environment that we're going to look at is a really interesting one: deep sea ocean vents. We finish our tour of the oceans by looking at a strange and wondrous place, the deep sea. Most of the deep sea is cold and dark, with very little life except for a few chemoheteroorganotrophs growing on stuff that just floats down from the surface. Holger Janisch and his research team at Woods Hole ventured down in 1977 to study deep sea ocean vents. Ocean vents appear at intersections of tectonic plates. Water seeps into the cracks, gets heated by magma, and picks up hydrogen sulfide, iron, sulfur, ammonia, methane, and H2. This water, which can be 5 to 400 degrees centigrade, then flows back into the ocean. They expected to find hot water and interesting geothermal formations. To their great surprise, an entire biologic community was found, including giant mussels, crabs, lobsters, and tube worms. What was the primary producer? It turns out it is a microbe living on hydrogen sulfide and oxygen. Remember, I said oxygen is available at the very deep sea. An overall schematic of a typical ocean vent is shown here. Note the large amount of reduced ions that are available for use. The seawater seeps into these areas of cracks where there's magma. It gets superheated and reacts and creates all these molecules shown here. These will spew out into the ocean and release all sorts of reducing chemicals. This can be extremely hot, up to 350 degrees centigrade, but it will quickly cool by the cool water around it, and so you'll get all sorts of different interesting life forms living in these areas. Lots of chemistries are available for bacteria to use and take advantage of. You note these are lots of reducing chemicals, so there's lots of electron donors, there's lots of electron acceptors available. And if this is true, you know there's going to be life forms and microorganisms and bacteria that take advantage of it. So, just thinking about what we said, what is available at the deep sea to use as a source of energy? The answers are both organic compounds and inorganic compounds. So A and C are correct, so you should say D. There are no Snickers bars. So, what do we find at the deep sea? Many of the microbes present in these ocean vents are living off reduced inorganic compounds. Here we have several examples of the various reactions that take place, yielding energy for these cells. All of them use carbon dioxide as their source of carbon. Note also that methanogens and methylotrophs are often found together. Why do you think that would be true? It's true because they actually live on each other's compounds, right? Products. Pyrodictium abyssi has an optimal growth temperature of 105 degrees centigrade. That's amazing, right? It is a hydrogen oxidizer, and it links this to sulfur reduction. Here are a couple more examples of the kind of organisms you can find in the deep sea. One of these is Geoglobus ahangari. It is in the phylum Euarchaeota, so it is an archaea. It is a motile coccus and thermophile growing from 65 to 90 degrees centigrade. It uses iron as its terminal electron acceptor. It can grow chemolithotrophically using hydrogen as in its electron donor. But it can also use acetate and many other organics. Thus, is an important acetate sink, meaning that it will take up the acetate and get rid of it or use it. Another example of an organism available in the deep sea is Thermococcus atlanticus. It is not a chemoheterolithotroph. It is a chemoheteroorganotroph. Thermococcus atlanticus is a very common organism at deep sea ocean vents and is an obligate thermophile, but a heterotroph, growing on proteinaceous substrates. Other species of the genus can use carbohydrates, and they have intense interest in biotechnology because they have enzymes that are thermal stable, but they grow on substrates that are not so corrosive to metal fermenters. A lot of the other organisms, like Geoglobus ahangari, will grow on hydrogen and iron. Those can be more difficult to deal with. The guys that use sulfur can be very corrosive, so that's also difficult to use. So they're interested in looking at this one. We end this little video by talking about tube worms, and this is one example of the organisms that are present in the deep sea. There are also many eukaryotic species down at these ocean vents. Note the crabs and lobsters in the video that I posted on Canvas that you can watch. These feed on microbes growing on surfaces. Much closer symbiotic relationships also form. One example is tube worms and their sulfur oxidizing bacteria contained inside them. The tube worm uses a type of hemoglobin with zinc instead of iron as the active ion. This delivers CO2 and hydrogen sulfide to the symbiont. The symbiont uses these in its metabolism. In return, the microbe makes organic matter for the worm. All right, and here ends our coverage of the microbial diversity of the environment. I hope you have a good sense and appreciation of all the microorganisms present in the environment.